Glutamate. In recent years, the neurotransmitter glutamate has attained a key theoretical role in the hypothesized pathophysiology of schizophrenia, as well as in a number of other psychiatric disorders, including depression. It is also now a key target of novel psychopharmacologic agents for future treatments of schizophrenia and depression. In order to understand theories about glutamate in schizophrenia and other psychiatric disorders, how the malfunctioning of glutamate systems impacts dopamine systems in schizophrenia, and how glutamate systems might become important targets of new therapeutic drugs for schizophrenia, it is necessary to review the regulation of glutamate neurotransmission. Glutamate is the major excitatory neurotransmitter in the central nervous system and sometimes considered to be the master switch of the brain, since it can excite and turn on virtually all CNS neurons. The synthesis, metabolism, receptor regulation, and key pathways of glutamate are therefore critical to the functioning of the brain and will be reviewed here. Glutamate synthesis. Glutamate or glutamic acid is a neurotransmitter which is an amino acid. Its predominant use is not as a neurotransmitter, but as an amino acid building block for protein biosynthesis. When used as a neurotransmitter, it is synthesized from glutamine in glia, which also assist in the recycling and regeneration of more glutamate following glutamate release during neurotransmission. When glutamate is released from synaptic vesicles stored within glutamate neurons, it interacts with receptors in the synapse and is then taken up into neighboring glia by a reuptake pump known as an excitatory amino acid transporter, EAAT, Figure 4 to 19a. The presynaptic glutamate neuron and the postsynaptic site of glutamate neurotransmission may also have EAATs, not shown in the figures. But these EAATs do not appear to play as important a role in glutamate recycling and regeneration as the EAATs in glia, Figure 4 to 19a. After reuptake into glia, glutamate is converted into glutamine inside the glia by an enzyme known as glutamine synthetase, arrow 3 in Figure 4 to 19b. It is possible that glutamate is not simply reused, but rather converted into glutamine, to keep it in a pool for neurotransmitter use, rather than being lost into the pool for protein synthesis. Glutamine is released from glia by reverse transport out of them by a pump or transporter known as a specific neutral amino acid transporter, SNAT, arrow 4 in Figure 4 to 19c. Glutamine may also be transported out of glia by a second transporter known as a glial alanine serine cysteine transporter or ASCT, not shown. When glial SNATs and ASCTs operate in the inward direction, they transport glutamine and other amino acids into glia. Here, they are reversed so that glutamine can get out of the glia and hop a ride into a neuron via a different type of neuronal SNAT, operating inwardly in a reuptake manner, arrow 5 in Figure 4 to 19c. Once inside the neuron, glutamine is converted back into glutamate for use as a neurotransmitter by an enzyme in mitochondria called glutaminase, arrow 6 in Figure 4 to 19d. Glutamate is then transported into synaptic vesicles via a vesicular glutamate transporter, VGLUT, arrow 7 in Figure 4 to 19d, where it is stored for subsequent release during neurotransmission. Once it is released, glutamate's actions are stopped not by enzymatic breakdown, as in other neurotransmitter systems, but by removal by EAATs on neurons or glia. And the whole cycle is started again, Figures 4 to 19a through d. Synthesis of glutamate CO transmitters. Glycine and D-serine glutamate systems are curious in that one of the key receptors for glutamate requires a co-transmitter in addition to glutamate in order to function. That receptor is the NMDA, N-methyl D-aspartate, receptor, described below, and the co-transmitter is either the amino acid glycine, Figure 4 to 20, or another amino acid closely related to glycine. known as D-serine, figure 4 to 21. Glycine is not known to be synthesized by glutamate neurons, so glutamate neurons must get the glycine they need for their NMDA receptors either from glycine neurons or from glia, figure 4 to 20. Glycine neurons release glycine, but they contribute only a small amount of glycine to glutamate synapses. Glycine is unable to diffuse very far from neighboring glycine neurons because the glycine they release is taken back up into those neurons by a type of glycine reuptake pump known as the type 2 glycine transporter or GlyT2, figure 4 to 20. Thus, neighboring glia are thought to be the source of most of the glycine available for glutamate synapses. Glycine itself can be taken up into glia as well as into glutamate neurons from the synapse by a type 1 glycine transporter or GlyT1, figure 4 to 20. Glycine can also be taken up into glia by a glial SNAT, specific neutral amino acid transporter. Glycine is not known to be stored within synaptic vesicles of glia, but as we will learn below, the companion neurotransmitter D-serine is thought possibly to be stored within some type of synaptic vesicle within glia. Glycine in the cytoplasm of glia is nevertheless somehow available for release into synapses, and it escapes from glial cells by riding outside them and into the glutamate synapse on a reversed GlyT1 transporter, figure 4 to 20. Once outside, glycine can get right back into the glia by an inwardly directed GlyT1, which functions as a reuptake pump and is the main mechanism responsible for terminating the action of synaptic glycine, figure 4 to 20. GlyT1 transporters are probably also located on the glutamate neuron, but any release or storage from the glutamate neuron is not well characterized, figure 4 to 20. Later, in Chapter 5, we will discuss novel treatments for schizophrenia that boost glycine action, and thus glutamate action, at NMDA receptors. Such treatments are in clinical testing and include inhibitors of the key glycine transporter GlyT1, called selective glycine reuptake inhibitors or SGRIs. Glycine can also be synthesized from the amino acid L-serine, derived from the extracellular space, bloodstream, and diet, transported into glial cells by an L-serine transporter, LSERT, and converted from L-serine into glycine by the glial enzyme serine hydroxymethyl transferase, SHMT, figure 4 to 20. This enzyme works in both directions, either converting L-serine into glycine or glycine into L-serine. How is the cotransmitter D-serine produced? D-serine is unusual in that it is a D-amino acid, whereas the 20 known essential amino acids are all L-amino acids, including D-serine's mirror image amino acid L-serine. It just so happens that D-serine has high affinity for the glycine site on NMDA receptors, and that glia are equipped with an enzyme called D-serine racemase that can convert regular L-serine into the neurotransmitting amino acid D-serine and vice versa, figure 4 to 21. Thus, D-serine can be derived either from glycine or from L-serine, both of which can be transported into glia by their own transporters, and then glycine converted to L-serine by the enzyme SHMT. And finally L-serine converted into D-serine by the enzyme D-serine racemase, figure 4 to 21. Interestingly, the D-serine so produced may be stored in some sort of vesicle in glia for subsequent release on a reversed glial D-serine transporter, DSERT, for neurotransmitting purposes at glutamate synapses containing NMDA receptors. D-serine's actions are not only terminated by synaptic reuptake via the inwardly acting glial DSERT, but also by an enzyme, D-amino acid oxidase, DAO, that converts D-serine into inactive hydroxypyruvate, figure 4 to 21. Below, we will discuss how the brain makes an activator of DAO, known not surprisingly as D-amino acid oxidase activator or DAOA. The gene that makes DAOA may be one of the important regulatory genes that contribute to the genetic basis of schizophrenia, as will be explained below in the section on the neurodevelopmental hypothesis of schizophrenia. Glutamate receptors. There are several types of glutamate receptors, Figure 4-22 and Table 4-7, including the neuronal presynaptic reuptake pump, EAAT, or excitatory amino acid transporter, and the vesicular transporter for glutamate into synaptic vesicles, VGLUT, both of which are types of receptors. The properties of various transporters are discussed in Chapter 2. Shown also on the presynaptic neuron as well as the postsynaptic neuron are metabotropic glutamate receptors, Figure 4-22. Metabotropic glutamate receptors are those glutamate receptors which are linked to G proteins. The properties of G protein-linked receptors are also discussed in Chapter 2. There are at least 8 subtypes of metabotropic glutamate receptors, organized into 3 separate groups, Table 4-7. Research suggests that Group 2 and Group 3 metabotropic receptors can occur presynaptically, where they function as autoreceptors to block glutamate release, Figure 4-23. Drugs that stimulate these presynaptic autoreceptors as agonists may therefore reduce glutamate release and be potentially useful as anticonvulsants and mood stabilizers, and may also protect against glutamate excitotoxicity, as will be explained below. Group 1 metabotropic glutamate receptors may be located predominantly postsynaptically, where they hypothetically interact with other postsynaptic glutamate receptors to facilitate and strengthen responses mediated by ligand-gated ion channel receptors for glutamate during excitatory glutamatergic neurotransmission, Figure 4-22. NMDA, N-methyl D-aspartate, AMPA, alpha-amino 3-hydroxy-5-methyl-4-isoxazole propionic acid, and kainate receptors for glutamate, named after the agonists that selectively bind to them, are all members of the ligand-gated ion channel family of receptors, Figure 4-22 and Table 4-7. These ligand-gated ion channels are also known as ionotropic receptors and ion channel-linked receptors. The properties of ligand-gated ion channels are discussed in Chapter 3. They tend to be postsynaptic and work together to modulate excitatory postsynaptic neurotransmission triggered by glutamate. Specifically, AMPA and kainate receptors may mediate fast excitatory neurotransmission, allowing sodium to enter the neuron to depolarize it, Figure 4-24. NMDA receptors in the resting state are normally blocked by magnesium, which plugs a calcium channel, Figure 4-25. NMDA receptors are an interesting type of coincidence detector that can open to let calcium into the neuron to trigger postsynaptic actions from glutamate neurotransmission only when 3 things occur at the same time. Glutamate occupies its binding site on the NMDA receptor, glycine or D-serine binds to its site on the NMDA receptor, and depolarization occurs, allowing the magnesium plug to be removed, Figures 4-25 and 4-26. Some of the many important signals by NMDA receptors that are activated when NMDA calcium channels are opened include long-term potentiation and synaptic plasticity, as will be explained later in this chapter. Key glutamate pathways in the brain. Glutamate is a ubiquitous excitatory neurotransmitter that seems to be able to excite nearly any neuron in the brain. That is why it is sometimes called the master switch. Nevertheless, there are about a half dozen specific glutamatergic pathways that are of particular relevance to psychopharmacology, and especially to the pathophysiology of schizophrenia, Figure 4-27. They are A. Cortica brainstem, B. Cortica striatal, C. Hippocampal striatal, D. Thalamocortical, E. Cortica thalamic, F. Cortica cortical, direct, G. Cortica cortical, indirect. A. Cortica brainstem glutamate pathways. A very important descending glutamatergic pathway projects from cortical pyramidal neurons to brainstem neurotransmitter centers, including the raphe for serotonin, the ventral tegmental area, VTA, and substantia nigra for dopamine, and the locus ceruleus for norepinephrine, pathway A in Figure 4 to 27. This pathway is the cortica brainstem glutamate pathway, and it is a key regulator of neurotransmitter release. Direct innervation of monoamine neurons in the brainstem by these excitatory cortica brainstem glutamate neurons stimulates neurotransmitter release, whereas indirect innervation of monoamine neurons by these excitatory cortica glutamate neurons via GABA interneurons in the brainstem blocks neurotransmitter release. B. Cortica striatal glutamate pathways. A second descending glutamatergic output from cortical pyramidal neurons projects to the striatal complex, pathway B in Figure 4 to 27. This pathway is known as the cortica striatal glutamate pathway when it projects to the dorsal striatum, or the cortica accumbens glutamate pathway when it projects to a specific area of the ventral striatum known as the nucleus accumbens. In either case, these descending glutamate pathways terminate on GABA neurons destined for a relay station in another part of the striatal complex called the globus pallidus. C. Hippocampal accumbens glutamate pathway. Another key glutamate pathway projects from the hippocampus to the nucleus accumbens and is known as the hippocampal accumbens glutamate pathway. See in Figure 4 to 27. Specific theories link this particular pathway to schizophrenia. See below. Like the cortica striatal and cortica accumbens glutamate pathways, B in Figure 4 to 27. The hippocampal glutamate projection to the nucleus accumbens also terminates on GABA neurons there that in turn project to a relay station in the globus pallidus. D. Thalamocortical glutamate pathway. This pathway, D in Figure 4 to 27, brings information from the thalamus back into the cortex, often to process sensory information. E. Cortica thalamic glutamate pathway. A fifth glutamate pathway, known as the cortica thalamic glutamate pathway, projects directly back to the thalamus, pathway E in Figure 4 to 27, where it may direct the manner in which neurons react to sensory information. F. Direct cortica cortical glutamate pathways. Finally, a complex of many cortica cortical glutamate pathways are present within the cortex, pathways F and G in Figure 4 to 27. On the one hand, pyramidal neurons can excite each other within the cerebral cortex via direct synaptic input from their own neurotransmitter glutamate, F in Figure 4 to 27. G. Indirect cortica cortical glutamate pathways. On the other hand, one pyramidal neuron can inhibit another via indirect input, namely via interneurons that release GABA, G in Figure 4 to 27. The NMDA hypofunction hypothesis of schizophrenia. Ketamine and phencyclidine, a major current hypothesis for the cause of schizophrenia, proposes that glutamate activity at NMDA receptors is hypofunctional due to abnormalities in the formation of glutamatergic NMDA synapses during neurodevelopment. This so-called NMDA receptor hypofunction hypothesis of schizophrenia arises in part from observations that when NMDA receptors are made hypofunctional by means of the NMDA receptor antagonists PCP, phencyclidine or ketamine, Figure 4 to 28, this produces a psychotic condition in normal humans very similar to symptoms of schizophrenia. Hypothetically, Genetic abnormalities also make NMDA receptors and their synapses hypofunctional in order to cause schizophrenia itself. Amphetamine, which releases dopamine, also produces a psychotic condition of delusions and hallucinations in normal humans similar to the positive symptoms of schizophrenia. What is so attractive about the NMDA receptor hypofunction hypothesis of schizophrenia is that unlike amphetamine, which activates only positive symptoms, PCP and ketamine also mimic the cognitive, negative, and affective symptoms of schizophrenia such as social withdrawal and executive dysfunction. Another attractive aspect of the NMDA hypofunction hypothesis is that it can also explain the dopamine hypothesis of schizophrenia, namely, as a downstream consequence of hypofunctioning NMDA receptors. The NMDA hypofunction hypothesis of schizophrenia. Faulty NMDA synapses on GABA interneurons in prefrontal cortex. Although NMDA receptors and synapses are ubiquitous throughout the brain, and PCP or ketamine block all of them. A current leading theory of schizophrenia suggests that schizophrenia may be caused by neurodevelopmental abnormalities in the formation of glutamate synapses at a specific site, namely, at certain GABA interneurons in the cerebral cortex, CG in Figure 4-27, and also Box 1 in both Figure 4-29A and Figure 4-29B. Something appears to be wrong with the genetic programming of those particular GABA interneurons that can be identified in prefrontal cortex as containing a calcium-binding protein called parvalbumin. Figure 4-29B. These parvalbumin-containing GABA interneurons appear to be faulty postsynaptic partners to incoming glutamate input from pyramidal neurons in prefrontal cortex, and to form defective NMDA receptor containing synaptic connections with incoming pyramidal neurons. Figure 4-29B, Box 1. Compare Figure 4-29A, Box 1. Thus, they have hypofunctioning NMDA receptors on their dendrites, defective synapses between the glutamate neuronal axons and the GABA interneuronal dendrites, and thus faulty glutamatergic information coming into the GABA interneuron. Figure 4-29B, Box 1. This so-called disconnectivity may be genetically programmed from a variety of faulty genes that all converge on the formation of these particular NMDA synapses. Parvalbumin-containing GABA interneurons in the prefrontal cortex of patients with schizophrenia have other problems as a consequence of this disconnectivity, in that they also have deficits in the enzyme that makes their own neurotransmitter GABA, namely, decreased activity of GAD67, glutamic acid decarboxylase, causing a compensatory increase in the postsynaptic amount of alpha-2 subunit containing GABAA receptors in the postsynaptic axon initial segment of the pyramidal neuron they innervate. Figure 4-29B. Box 2. Compare Figure 4-29A, Box 2. What are the consequences of the hypothetical disconnectivity of glutamate with these particular GABA interneurons? When parvalbumin-containing GABA interneurons fail to function properly, they do not adequately inhibit key glutamatergic pyramidal neurons in the prefrontal cortex, causing those glutamate neurons to become hyperactive. Figure 4-29B, Box 3. Compare Figure 4-29A, Box 3. This hypothetically disrupts the functioning of downstream neurons, especially dopamine neurons. Figures 4-30B, 4-31B, and 4-32B, explained below. So, one sick synapse in a neuronal circuit can affect the whole circuit. From GABA interneuron and the glutamate neurons it innervates, to downstream dopamine neurons and beyond. Linking the NMDA hypofunction hypothesis of schizophrenia with the dopamine hypothesis of schizophrenia. Positive symptoms. A complex set of interactions allows glutamate to determine dopamine release. Most relevant to schizophrenia are the glutamate pathways that regulate the mesolimbic and mesocortical dopamine pathways shown in figures 4 to 11 through 4 to 16. Cortico-brainstem glutamate pathways regulate the output of glutamate from the cortex to the brainstem neurotransmitter center known as the ventral tegmental area, VTA, for both the mesolimbic dopamine projection, pathway A in figure 4 to 27 and in figure 4 to 30A, and for the mesocortical dopamine projections, pathway A in figure 4 to 27 and in figure 4 to 32A. First, we will discuss the glutamate regulation of mesolimbic dopamine neurons, figure 4 to 30. It appears that the cortico-brainstem glutamate neurons that innervate only the dopamine neurons projecting from the VTA to the nucleus accumbens, i.e., the mesolimbic dopamine pathway, directly innervate those specific dopamine neurons, figure 4 to 30A, and thus stimulate them. You can imagine what would happen if these upstream glutamate neurons were too active, figures 4 to 29B and 4 to 30B. They would cause hyperactivity of the downstream mesolimbic dopamine neurons, figure 4 to 30B. This is exactly what is hypothesized to be happening in schizophrenia. The dopamine hyperactivity of these downstream mesolimbic dopamine neurons is associated with the positive symptoms of schizophrenia but is actually caused hypothetically by disconnectivity in upstream glutamate neurons, namely, defective and hypofunctional neurodevelopmental glutamate innervation of parvalbumin containing GABA interneurons at NMDA receptor containing synapses, figure 4 to 29B and 4 to 30B. It is also possible that the disconnectivity of upstream glutamate neurons in the hippocampus contributes to downstream mesolimbic dopamine hyperactivity via a 4-neuron circuit, figure 4 to 31A. That circuit consists of 1. The disconnected and defective hippocampal parvalbumin containing GABA interneuron, going to 2. The hippocampal glutamate neuron projecting to the nucleus accumbens. Then that neuron projecting to 2 GABA spiny neurons in sequence, 3. The first GABA spiny neuron going from nucleus accumbens to globus pallidus. And finally, 4. The second GABA spiny neuron going from globus pallidus to VTA, figure 4 to 31A. Loss of adequate glutamate function at parvalbumin containing GABA interneurons in the hippocampus could lead to hyperactive glutamate output from glutamate neurons that project by this circuit to the mesolimbic dopamine neurons in the VTA, with consequential dopamine hyperactivity and positive symptoms of schizophrenia, figure 4 to 31B. Stimulating 2 GABA neurons in sequence has the net effect of disinhibition, inhibition of inhibition, at the VTA. The same result as direct stimulation, which was illustrated for the prefrontal cortex in figure 4 to 30A. The bottom line is that excessive upstream glutamate output from either the prefrontal cortex or the hippocampus may contribute to downstream dopamine hyperactivity and positive symptoms of schizophrenia. Linking the NMDA hypofunction hypothesis of schizophrenia with the dopamine hypothesis of schizophrenia. Negative symptoms next. We will discuss the glutamate regulation of mesocortical dopamine neurons, figure 4 to 32. It appears that different cortico-brainstem glutamate neurons regulate those unique dopamine neurons in the VTA that project only to the prefrontal cortex, the mesocortical dopamine pathway, figure 4 to 32A, then regulate those dopamine neurons in the VTA that project to the nucleus accumbens as the mesolimbic dopamine pathway, figure 4 to 30A. Thus, different populations of glutamate neurons regulate the different populations of dopamine neurons. Cortico-brainstem glutamate neurons destined to regulate mesocortical dopamine neurons in the VTA do not directly innervate them, figure 4 to 32A, as do the cortico-brainstem glutamate neurons destined to regulate mesolimbic dopamine neurons in the VTA, figure 4 to 30a. Instead, the glutamate neurons regulating mesocortical dopamine neurons do so by indirectly innervating an inhibitory GABA interneuron that itself innervates the mesocortical dopamine neurons, figure 4 to 32a. Thus, activation of these particular glutamate neurons leads first to activation of GABA interneurons, which then inhibit mesocortical dopamine neurons, figure 4 to 32a. You can imagine what would happen if these glutamate neurons were too active, figure 4 to 29b and 4 to 32b. Hypoactivity of the mesocortical dopamine neurons, figure 4 to 31b. This is exactly what is hypothesized to be occurring in schizophrenia. The dopamine hypoactivity of these mesocortical dopamine neurons is associated with the negative and cognitive symptoms of schizophrenia. It is hypothetically caused by the same upstream disconnectivity of glutamate with GABA interneurons that causes the hyperactivity of mesolimbic dopamine neurons, namely the neurodevelopmental abnormality in glutamate innervation of parvalbum in containing GABA interneurons at their NMDA synapses, figures 4 to 29b and 4 to 30b. Only in this case, it is affecting a different population of glutamate neurons in the prefrontal cortex and with different downstream consequences, namely production of negative and cognitive symptoms of schizophrenia rather than positive symptoms. Different populations of cortica brainstem glutamate projections thus regulate the release of dopamine from both the mesocortical and the mesolimbic dopamine projections. Although it appears that this regulation is the opposite for the glutamate neurons that regulate the mesolimbic dopamine pathway compared with the glutamate neurons that regulate the mesocortical dopamine pathway, compare figures 4 to 30a and 4 to 32a. All due to the presence or absence of a GABA interneuron in the VTA. Neurodevelopment and genetics in schizophrenia. What causes schizophrenia? Nature, i.e., genetics, or nurture, i.e., the environment or epigenetics. The modern answer seems to be both. Modern theories of schizophrenia no longer propose that a single gene causes schizophrenia, pure nature, figure 4 to 33, any more than a bad mother can cause schizophrenia, pure nurture. Instead, it seems more likely that schizophrenia is a conspiracy among many genes and many environmental stressors to cause abnormal development of brain connections throughout life. In fact, not only is there no single gene for schizophrenia, or for any other major psychiatric disorder, figure 4 to 33, there is no single gene for any specific psychiatric symptoms, behaviors, personalities, or temperaments, figure 4 to 34. Genes do not code for mental illnesses or for psychiatric symptoms. Instead, genes code for proteins, figure 4 to 35. Today, mental illnesses are thought to be linked in part to someone inheriting an entire portfolio of many genes that carry risk for a mental illness, especially in combination, and set the stage for a mental illness, but do not cause mental illness per se, figure 4 to 36. In schizophrenia, multiple risk genes each hypothetically code for a subtle molecular abnormality, figure 4 to 35. Any one of which alone may be clinically silent until stress from the environment puts a load on these defective genes, and also causes even normal genes to be expressed when they should be silenced, or silenced when they should be expressed, figure 4 to 36, a process called epigenetics, discussed briefly in Chapter 1, figure 1 to 30. Thus, mental illnesses are due not only to genes that are abnormal in their DNA and in the function of the proteins they code, but also to normal genes that make normal functioning proteins but are activated or silenced at the wrong times by the environment, nature and nurture, figure 4 to 36. In the case of schizophrenia, The problem seems to be disconnectivity of neurons, particularly in hippocampus and prefrontal cortex, and particularly at glutamate synapses with NMDA receptors that become hypofunctional. Stress, traumatic experiences, learning, sensory experiences, sleep deprivation, toxins, and drugs are all examples of how normal genes, such as those that regulate the formation and removal of synapses, are turned on and off by the environment. Figure 4-36. Cannabis use is a particularly malicious environmental stressor to those vulnerable to schizophrenia. These are all examples of the notion of experience-dependent development of synaptic connections, something that is hypothetically abnormal in schizophrenia, both from the experiences that the patient may have and from the genes that respond to these experiences. Thus, in schizophrenia, an individual not only hypothetically inherits many abnormal genes, which may converge on the formation of NMDA receptor containing glutamate synapses, but theoretically also has notable experiences from a stressful environment that cause the abnormal expression or abnormal silencing of perfectly normal genes in just the necessary sequence to cause this illness. Figure 4-36. The best evidence that the environment is involved in schizophrenia is that only half of identical twins of patients with schizophrenia also have schizophrenia. Having identical genes is thus not enough to cause schizophrenia, but presumably epigenetics is also in play such that the affected twin not only expresses some abnormal genes that the unaffected twin might not express, but also expresses some normal genes at the wrong time and silences other normal genes at the wrong time, and together these factors cause schizophrenia in one twin but not the other. The best evidence for the role of disconnectivity genes in schizophrenia is the convergence of evidence implicating multiple genes that regulate not only neuronal connectivity in general, but glutamate synapse formation and removal in particular. Table 4-8. This includes dysbindin, neuregulin, ERBB4, and DISC1, among others. Figures 4-36 and 4-37. Dysbindin, also known as dystrobrevin binding protein 1, is involved in the formation of synaptic structures and regulation of the activity of the vesicular transporter for glutamate, VGLUT. Neuregulin is involved in neuronal migration, genesis of glial cells, and subsequent myelination of neurons by glia. Neuregulin also activates an ERBB4 signaling system that is co-localized with NMDA receptors. These ERBB4 receptors also interact with the postsynaptic density of glutamate synapses and may be involved in mediating the neuroplasticity triggered by NMDA receptors. Both dysbindin and neuregulin impact the formation and function of the postsynaptic density, a set of proteins that interacts with the postsynaptic membrane to provide both structural and functional regulatory elements for neurotransmission and for NMDA receptors. DISC1, disrupted in schizophrenia 1, is aptly named for a disrupted gene linked to schizophrenia that makes a protein involved in neurogenesis, neuronal migration, and dendritic organization, and also affects the transport of synaptic vesicles into presynaptic glutamate nerve terminals and regulates CAMP signaling, which would affect the functions of glutamate neurotransmission mediated by metabotropic glutamate receptors. Dysbindin, DISC1, and neuregulin all affect normal synapse formation. They all affect NMDA receptor number by altering NMDA receptor trafficking to the postsynaptic membrane, NMDA receptor tethering within that membrane, and NMDA receptor endocytosis that cycles receptors out of the postsynaptic membrane to remove them. Thus, it is easy to see how multiple genetic or epigenetic abnormalities in the expression of these particular genes could lead to disconnectivity of glutamate neurons in schizophrenia. Figures 4-37 and 4 to 38. Other risk genes implicate specific proteins that directly regulate glutamate synapses and, if abnormally expressed, could add to the misery of a disconnected and dysfunctional NMDA glutamate synapse. Figure 4 to 38. For example, the gene for DAOA, D-amino acid oxidase activator, codes for a protein that activates the enzyme DAO, D-amino acid oxidase. DAO degrades the co-transmitter D-serine that acts at glutamate synapses and at NMDA receptors. DAOA activates this DAO enzyme. So abnormalities in the gene for DAOA would be expected to alter the metabolism of D-serine. This in turn would alter glutamate neurotransmission at NMDA receptors. Another schizophrenia susceptibility gene active directly at glutamate synapses is RSG4, regulator of G protein signaling. And this gene product also impacts metabotropic glutamate receptor signaling through the G protein-coupled signal transduction system. Normally, when glutamate synapses are active, their NMDA receptors trigger an electrical phenomenon known as long-term potentiation, LTP. With the help of dysbindin, DISC1, and neuregulin, LTP leads to structural and functional changes of the synapse that make neurotransmission more efficient, sometimes called strengthening of synapses. Figure 4 to 39. This includes increasing the number of AMPA receptors. AMPA receptors are important for mediating excitatory neurotransmission and depolarization at glutamate synapses. Thus, more AMPA receptors can mean a strengthened synapse. Synaptic connections that are frequently used develop recurrent LTP and consequential robust neuroplastic influences, thus strengthening them according to the old saying, nerves that fire together wire together. However, if something is wrong with the genes that regulate synaptic strengthening, it is possible that this causes less effective use of these synapses, makes the NMDA receptors hypoactive, leading to ineffective LTP and fewer AMPA receptors trafficking into the postsynaptic neuron. Figure 4 to 39. Such a synapse would be weak, theoretically causing inefficient information processing in its circuit and possibly also causing symptoms of schizophrenia. The strengthening or weakening of a glutamate synapse is an example of activity-dependent, or use-dependent, or experience-dependent regulation of NMDA receptors and functionality at glutamate synapses. This not only occurs when these synapses first form, but continues throughout life as a sort of ongoing remodeling in response to what experiences the individual has, and thus how much that synapse is used or neglected. Abnormalities in these continuing dynamics at NMDA receptors and glutamate synapses may explain why the course of schizophrenia is progressive and changes over time for most patients, namely, from an asymptomatic period to a prodrome, to a first-break psychosis with robust treatment responsiveness, to multiple psychotic episodes with declining treatment responsiveness, to a state of pervasive negative and cognitive symptoms without recovery. Another important aspect of synaptic strength is that it likely determines whether a given synapse is eliminated or maintained. Specifically, strong synapses with efficient NMDA neurotransmission and many AMPA receptors survive, whereas weak synapses with few AMPA receptors may be targets for elimination. Figure 4 to 39. This normally shapes the brain's circuits so that the most critical synapses are not only strengthened but also survive the ongoing selection process, keeping the most efficient and most frequently utilized synapses, while eliminating inefficient and rarely utilized synapses. However, if critical synapses are not adequately strengthened in schizophrenia, it could lead to their wrongful elimination, causing disconnectivity that disrupts information flow from circuits now deprived of synaptic connections where communication needs to be efficient. Figure 4 to 39. Competitive elimination of weak but critical synapses during adolescence could even explain why schizophrenia has onset at this time. Normally, almost half of the brain's synapses are eliminated in adolescence, figure 4 to 40. In adulthood, you may lose and replace elsewhere about 7% of the synapses in your cortex every week. If abnormalities in genes for dysbindin, neuregulin, and/or DISC1 lead to the lack of critical synapses being strengthened, these critical synapses may be mistakenly eliminated during adolescence with disastrous consequences, namely the onset of symptoms of schizophrenia. Also, it may be that the disconnectivity of abnormal glutamate synapses present from birth is masked by the presence of many additional weak connections prior to adolescence. Acting with exuberance to compensate for defective glutamate connectivity, and with that compensation destroyed by the normal competitive elimination of synapses in adolescence, schizophrenia emerges. Neuroimaging circuits in schizophrenia. Imaging brain circuits in patients with schizophrenia with functional magnetic resonance imaging, fMRI, uncovers abnormal information processing in brain areas linked to cognition and emotion. Modern psychiatric research techniques can put a load on brain circuits and thereby perform a type of psychiatric stress test while visualizing the activity of the brain circuits. fMRI brain scanners can most readily detect the activity of neurons near the surface of the brain, which are mostly pyramidal neurons in the cortex. Although some deeper gray matter areas such as the striatum and amygdala can also be imaged. The activity of neurons so visualized in the cortex is the first leg of various brain circuits. Especially glutamate neurons, as shown in figure 4 to 27, acting within feedback loops from the cortex to the striatal complex, with that information relayed to the thalamus via a GABA neuron. Then back to the cortex again via another glutamate neuron as cortico-striato-thalamo-cortical or CSTC feedback loops. Brain circuits like this are information processing engines that are activated by various tasks or loads placed upon them, and seeing them light up may be literally watching the brain think. Function in the brain is topographical, meaning different brain circuits process different kinds of information. For example, the dorsolateral prefrontal cortex, DLPFC, is thought to be most closely linked to cognitive functioning such as problem solving, whereas the ventromedial prefrontal cortex, VMPFC, along with the amygdala, is thought to be most closely linked to emotional functioning, such as mood. Neurons in various brain areas, stressed with an information processing load, literally light up a specific brain area that can be visualized with current neuroimaging techniques. Thus, doing a calculation can light up the DLPFC, and seeing a sad face can activate the VMPFC and amygdala. Studies in patients with schizophrenia suggest that they cannot adequately recruit the hippocampus during memory recall, even though output from the hippocampus seems to be high in the first place. Also, patients with schizophrenia do not seem to be able to appropriately activate the DLPFC during a working memory task. Compare figures 4 to 41a and 4 to 41b. With decreased recruitment correlated with worsening cognitive symptoms, figure 4 to 41c. Actually, the results are somewhat inconsistent across studies. And it appears that prefrontal cortical or hippocampal dysfunction in schizophrenia is likely to be more complicated than just up hyperactivation or down hypoactivation, but might be better characterized as out of tune. According to this concept, either too much or too little activation of neuronal activity in the prefrontal cortex is suboptimal and can potentially be symptomatic. Just as a guitar string is out of tune whether it has too much or too little tension on it. How can circuits in schizophrenia be both hyperactive and hypoactive? Patients with schizophrenia appear to utilize greater prefrontal resources when performing cognitive tasks and yet achieve lower accuracy because they have cognitive impairment despite their best efforts to perform near normal. Patients with schizophrenia engage the DLPFC, but do so inefficiently, recruiting greater neural resources and hyperactivating the DLPFC. When performing poorly, schizophrenia patients do not appropriately engage and sustain the DLPFC and thus show hypoactivation. Thus, DLPFC circuits in schizophrenia patients can either be underactive and hypofrontal or overactive and inefficient. It is interesting to note that unaffected siblings of patients with schizophrenia may have the very same inefficient information processing in DLPFC that schizophrenia patients have. Although unaffected siblings of schizophrenia patients might have some mild degree of cognitive impairment, they do not share the full syndrome of schizophrenia. However, neuroimaging reveals that they may share the same inefficient DLPFC functioning while performing cognitive tasks that characterizes their sibling with schizophrenia. The unaffected siblings of a schizophrenia patient may thus share some of the susceptibility genes for schizophrenia with their affected sibling, but not enough of these risk genes to have the full syndrome of schizophrenia itself. Functional neuroimaging also has the potential of unmasking inefficient information processing in clinically silent presymptomatic patients destined to progress to the full schizophrenia syndrome. But much further research is required to see if this will become clinically useful. Schizophrenia has also long been recognized as having impairments in the ability to identify and accurately interpret emotions from overt sources, including facial expressions. This may be due to inefficient information processing within the VMPFC and amygdala and can be measured by imaging the response of the amygdala to emotional input, especially from facial expressions. The amygdala is normally activated by looking at scary, threatening faces, or by assessing how happy or sad a face may be and while attempting to match emotions to faces, figure 4 to 42. Whereas healthy controls may activate the amygdala in response to scary or fearful or emotionally charged faces, figure 4 to 42a. Patients with schizophrenia may not, figure 4 to 42b. This may represent distortion of reality as well as impairment in recognizing negative emotions and in decoding negative emotions in schizophrenia. Failure to mount the normal, emotional response to a scary face can also represent an inability to interpret social cues and lead to distortions in judgment and reasoning in schizophrenia. Thus, these negative and affective symptoms of schizophrenia may be due in part to lack of emotional processing under circumstances when this should be occurring. On the other hand, a neutral face or neutral stimulus may provoke little activation of the amygdala in a healthy person, figure 4 to 43a, yet an overreaction in a patient with schizophrenia, figure 4 to 43b, who may mistakenly judge people negatively or conclude wrongly that another holds strong unfavorable impressions of them or may even be threatening them. Activating emotional processing in the amygdala when it is inappropriate may accompany the symptom of paranoia and lead to impaired interpersonal functioning, including problems in social communication. Thus, patients with schizophrenia may exhibit deficits in recognizing emotions that may be manifested either as positive or negative symptoms of this disorder. The underlying biological endophenotype of amygdala activation, or lack of activation, can be assessed with neuroimaging whether the patient is experiencing these symptoms or not. Looking at the efficiency of emotional information processing may help clinicians identify and understand emotional symptoms that are difficult for patients with schizophrenia to express. Imaging genetics and epistasis. Not only can the impact of a mental illness such as schizophrenia be imaged today, so can the impact of certain genes. That is, single genes can alter the efficiency of information processing in everyone, and as such may endow risk for mental illness, but not cause mental illness by themselves. As discussed above, figure 4 to 36. Thus, individuals with the gene for catechol-O-methyltransferase, COMT, that has higher enzyme activity, called VAL, for the amino acid valine substituted at a critical site, have lower dopamine levels in DLPFC, and thus less efficient information processing there compared to individuals with the gene for COMT with lower enzyme activity and higher dopamine levels, called MET, figure 4 to 44. This difference in neuronal effort is usually not apparent as cognitive difficulties in the normal population, but may serve as one of many risk factors for schizophrenia. Table 4 to 8 and figure 4 to 38. Don't have T with VAL. The effects of two or more risk genes working together to increase the risk of schizophrenia can now be demonstrated with neuroimaging that shows how certain risk genes conspire to decrease the efficiency of information processing in the DLPFC during a cognitive load in schizophrenia. Figure 4 to 44. That is, the VAL variant of COMT by itself may or may not consistently alter DLPFC activity during a working memory test in schizophrenia compared to individuals with the MET variant of COMT. But when combined with another genetic variant of another enzyme that independently reduces the availability of dopamine in prefrontal cortex, there is a more robust increase in DLPFC activity during a working memory load. Figure 4 to 44. That variant is the T form of the enzyme MTHFR, methylene tetrahydrofolate reductase, involved in the formation of dopamine and in regulating the activity of COMT at the level of gene expression. There can be reduced efficiency of information processing during a working memory load with the T form of MTHFR alone, but combined with the VAL form of COMT. The change in information processing is more robust, thus demonstrating the epistatic interaction of MTHFR-T with COMT-VAL, figure 4 to 44. So, you may want to have T with Valerie, but you don't necessarily want to have T with VAL, or your cognitive difficulties may be epistatically worsened if you have schizophrenia. Be careful of having your alphabet soup. Another example of epistasis is how the efficiency of information processing in DLPFC gets worse if you combine 2 or 3 of several of the schizophrenia risk genes that have horrible sounding names from various letters of the alphabet. NRG1, Neuregulin, ERBB4, and AKT. We have already mentioned above that NRG1 is involved in neuronal migration and synapse formation, and can impact the formation and function of the postsynaptic density, a set of proteins that interacts with the postsynaptic membrane to provide both structural and functional regulatory elements for neurotransmission and for NMDA receptors. NRG1 also affects NMDA receptor number by altering NMDA receptor trafficking to the postsynaptic membrane, NMDA receptor tethering within that membrane, and NMDA receptor endocytosis that cycles receptors out of the postsynaptic membrane to remove them. NRG1 directly interacts with ERBB4 and activates the ERBB4 signaling system that is co-localized with NMDA receptors and interacts with the postsynaptic density of glutamate synapses and is involved in mediating the neuroplasticity triggered by NMDA receptors. Thus, it is easy to see how a genetic abnormality in the NRG1 protein could readily interact in an undesirable way with a genetic abnormality in its receptor ERBB4. To cause problems with information processing and to increase the risk for schizophrenia. In fact, when patients with schizophrenia have the risk genes both for NRG1 and for ERBB4, but not when they have just one of them. Their information processing in DLPFC is abnormal, figure 4 to 44. How about AKT? AKT is a kinase enzyme, part of the intracellular signal transduction cascades discussed in Chapter 1, figures 1 to 9, 1 to 16, 1 to 18, 1 to 28. This particular kinase specifically interacts with beta-arrestin 2 and GSK3 during signal transduction and has been shown to regulate neuronal cell size and cell survival, and to increase synaptic plasticity. Malfunctioning of AKT can lead to overactivation of GSK3 and lack of internalization of dopamine D2 receptors, contributing to dopamine hyperactivity and the risk of schizophrenia, by itself or in combination with the risk gene for either NRG1 or ERBB4. The risk gene for AKT does not appear to compromise the efficiency of information processing in the DLPFC of patients with schizophrenia, figure 4 to 44. However, when combined with both the risk genes for NRG1 and ERBB4, the risk gene for AKT further increases the activation and inefficiency of information processing in DLPFC of patients with schizophrenia who have both the risk genes for NRG1 plus ERBB4. A further example of epistasis, but here among 3 risk genes, figure 4 to 44. Thus, it is apparent that the more risk genes, the more the bias towards circuit breakdown in schizophrenia, and the more likely the illness will manifest, as shown in the hypothetical interactions of figures 4 to 36 and 4 to 38. Summary: this chapter has provided a clinical description of psychosis, with special emphasis on the psychotic illness schizophrenia. We have explained the dopamine hypothesis of schizophrenia, and the related NMDA receptor hypofunction hypothesis of schizophrenia, which are the major hypotheses for explaining the mechanism for the symptoms of schizophrenia. The major dopamine pathways and the major glutamate pathways in the brain have all been described. Overactivity of the mesolimbic dopamine system may mediate the positive symptoms of psychosis and may be linked to hypofunctioning NMDA glutamate receptors in parvalbumin-containing GABA interneurons in the prefrontal cortex and hippocampus. Underactivity of the mesocortical dopamine system may mediate the negative, cognitive, and affective symptoms of schizophrenia and could also be linked to hypofunctioning NMDA receptors at different GABA interneurons. The synthesis, metabolism, reuptake, and receptors for both dopamine and glutamate are described in this chapter. Dopamine D2 receptors are targets of all known antipsychotic drugs. NMDA glutamate receptors require interaction not only with the neurotransmitter glutamate, but also with the CO transmitters glycine or D-serine. Disconnectivity of NMDA receptor containing synapses caused by genetic and environmental epigenetic influences is a major hypothesis for the cause of schizophrenia, including its upstream glutamate hyperactivity and NMDA receptor hypofunction, as well as its downstream increases in mesolimbic dopamine but decreases in mesocortical dopamine. A whole host of susceptibility genes that regulate neuronal connectivity and synapse formation also increase the risk for schizophrenia and converge upon the NMDA receptor containing glutamate synapse. As a hypothetical central biological flaw in schizophrenia, malfunctioning neural circuits can be imaged in schizophrenic patients, including those in the dorsolateral prefrontal cortex that are linked to cognitive symptoms, and those in the amygdala that are linked to symptoms of emotional dysregulation. The effects of risk genes on the efficient functioning of brain circuits can also be imaged, including the epistatic interaction of 2 or more risk genes.